Gentlemen, dear colleagues, first of all, I am very much pleased to see all of you here, and it is my pleasure to see the colleagues which I saw last year. A special pleasure to see new, young and very young faces of newcomers. I thank the organizing committee for inviting me, and of course the most of my thanks go to Olena Fesenko, who organized all this, almost everything. I think that despite the fact that last year I presented a talk about infrared spectroscopy of gold nanoparticles and proteins attached to them and some physical and chemical relationships, this time I decided to turn to another topic, and I think that I will give you a bit of rest from complicated physics, from complicated mathematical equations. This material will be somewhat simple, but I think it is very illustrative in the sense of obtaining knowledge about different mechanisms which underlie the processes of the formation of nanoparticles by living systems. This is a very important and very new and very well developing area of research and applications as well. So my talk will be devoted to the synthesis of nanoparticles, different nanoparticles, on the example of gold and selenium nanoparticles by bacterial cells. As you can see, I represent a purely biological institute. So I'm a physical chemist, but and I've been working at this institute for 20 years already. But this doesn't mean that I'm an alien in this institute. I'm not an Englishman in New York. I'm a physical chemist in a biological institute, and this is my pleasure to work there because this is a special interest to develop physicochemical approaches to studying biological systems. This is, to my mind, this is the right way to understand the very complicated processes. My task at this institute has been, since the beginning of my work, was to develop and apply spectroscopic approaches to studying these processes by specific interactions, biocomplexes, complicated biocomplexes, from simple complexes to enzymes, metalloenzymes, metalloproteins. So let's stick to biology and chemistry. I will give you a generalized overview of mechanisms which could be involved in the formation of nanoparticles by bacteria. In general, there are two very general mechanisms which could be involved. The first one, this is so to say purely chemical mechanism, which is, I would like to note it as biologically induced mechanism. It is involved in the fact when an element is reduced by cell components or secondary metabolites of living cells which have already been released or synthesized by a cell. It means that the cell may be living or may be dead. But in this case, the chemical processes involving these substances of biological origin take place. And this mechanism could be easily called chemical. But of course, without the cell, it wouldn't be able to be developed. So this is the first type of general mechanism, and as an example we will see this kind of mechanism, the reduction of gold-3 in the form of gold chloride complexes by bacterial cells, and I will try to show you that this process takes place according to general chemical mechanism. The second mechanism, which I will illustrate by another example, is the mechanism when an element is reduced by cellular metabolic redox activity, and it cannot proceed without a living cell. Of course, you can easily understand that if you find a nanoparticle or an ensemble of nanoparticles attached to the cell surface, it could be, according to both mechanisms, it could be reduced by a living cell, it could be reduced by a dead cell. But if you can find nanoparticles inside the cell, it most probably it can most probably be interpreted as a result of an activity of a living cell, because either the initial substances should transform from the surface inside the cell, and then nanoparticles could be formed there, or the living cell should reduce somehow the element, form nanoparticles, and then nanoparticles could be transferred into the cell through the membrane. It requires, of course, a metabolic activity of a cell, so the cell should be living. So in this case, we cannot say that this is a purely chemical mechanism, although, as all biology, this mechanism is related to chemical processes, of course. So all biology is a very complicated chemistry. As a chemist, I am sure of that. So this is a biochemical mechanism. And as an example, I will try to show you, to convince you, that this example, the reduction of selenium anions, in this case this is selenite, where selenium is the oxidation state +4, to selenium, and in particular to the formation of selenium nanoparticles, which have a lot of possible applications, and the process is rather interesting. Okay, so I will stick to the model bacterium. This bacterium is studied over the world, especially the bacteria of the genus Azospirillum. This species is very well studied. A lot of publications appear every year. And I will say a few words about this bacterium. Our institute studies the Azospirillum bacteria for 20 years already, or about 30 years. These bacteria are studied for over 35 years, and still an interest grows. A few words about this bacterium. It is interesting from many points of view. First of all, these bacteria are ubiquitous. It means that although it is called Azospirillum brasilense, for example, I speak about this species as Azospirillum brasilense, it was opened in Brazil, as you can understand, by a Brazilian researcher. Still you can find this bacterium out there in this grass outside. I'm sure of that, because in many climatic zones you can find this bacterium, from the North Pole to the very southern regions, and to Brazil, of course. So in Saratov we have got a lot of strains of this bacterium in our grasses, in our forests. This is a plant growth promoting bacterium. It is called plant growth promoting rhizobacterium, PGPR. This is a very common abbreviation. You can use it for your search, literature search. Plant growth promoting, it means that they show phytostimulating potential, and this is one of the interests why these bacteria are studied widely, because they help to plant crops, and sometimes currently a lot of biofertilizers are produced of these bacteria and sold already, although the effects from them are not straightforward. Sometimes the effects do not appear, and this is a big problem to make a study of the reasons for that. That is why they are still being studied. The plant growth promoting effects are because of phytohormone production and excretion out of the cell. These are soil bacteria. Atmospheric nitrogen fixation, which is very important not only for the plants, which get the nitrogen fixed from the atmosphere, but mostly for the bacteria itself, because in the absence of nitrogen nutrition, they can fix nitrogen and supply nitrogen compounds, first of all ammonium, to the cell metabolism, and if excess of nitrogen is fixed, then the plant to which it is associated can get this nitrogen as well. Some soil bioremediation properties are also applicable to these bacteria. For example, some of them are very stable to heavy metal stress. This means that plants could grow better in the presence of these bacteria, even in heavily polluted soils. Some of them can degrade oil pollution and some other organic pollution. So this is a very positive, a very beneficial trait of this bacterium. They can, as I have said already, they can be used as inoculants and co-inoculants in crop planting. They have been studied for 35 years almost. This is a simple image of these bacteria. Sometimes they are a bit prolonged, of the size of about 1 micrometer. There are several reviews which are published regularly, and in this year already a couple of reviews, including azospirillum, have also appeared. Okay, so let's stick to the formation of nanoparticles. This is a general picture. I should say that all the material which I will speak about is our own results. I decided to do like that, probably because it is more illustrative than everybody can take everybody's material and show as a review. I prefer to give examples which we study. Sometimes it is not yet totally understood, but to me it is more interesting and maybe more illustrative for you. So every material is ours, obtained in our laboratory. So this is an example of two strains of the same bacteria. I should say that these two strains sometimes differ greatly in the ecological behavior. That is why they are often compared as to the behavior of these strains in the nature. And sometimes they have different behavior, different processes, and different responses to the same ecological factors. This is very interesting from the viewpoint of how the same species but different strains, which occupy different ecological niches in the rhizosphere of plant roots, how they respond differently to the same stresses, to the same environmental effects. This strain is endophytic. Endophytes are capable of penetrating into the plant tissues. And to some extent, they are protected by plant tissues from the stresses, for example, from the pollution in the soil, from the drought stress, and from the lack of nutrients and so on. This strain is epiphytic. It means that it always colonizes the surface of roots. This is the next ecological niche, and that is why they could be always compared. In this case, when these bacteria were cultured in the medium containing this substance, orochloric acid, the oxidation state of gold here is +3. They reduce gold and they form gold nanoparticles. This is slightly larger because this is 500 nanometers. He's slightly lower, but in these two cases, the nanoparticles are very similar. They are spherical. The conditions, of course, play a role, but what I would like to compare. In this case, the nanoparticles are nanospheres in both cases. But in order to see whether the surface structure and composition play a role in the processes of the formation of nanoparticles, we took a special mutant strain. This is a very interesting mutant strain of this bacterium. One of its features is that the surface composition and structure is altered dramatically. It has been proven in our microbiology laboratory of our institute, so we took the strain from them and studied it. And we I hope it is clearly seen for everybody here, but for those who cannot see this left bottom side of the slide, the nanoparticles which are formed on the surface of this strain show very different shape. These are so-called nanoflowers. These are not aggregates of smaller particles, but these are really nanoflowers, and the careful examination by transmission electron microscopy shows that this is really so. So in this case, first of all, we showed that the structure and composition of the surface polysaccharides and surface biopolymers of the bacteria play a crucial role in the formation of specific shape of nanoparticles, and this could have a lot of very important applications. For example, nanoflowers, as many physicists and chemists working in this field know, have very specific plasmonic properties, and their synthesis sometimes is not that straightforward, not that simple. And in this case, it can be a very simple and straightforward route to synthesize such nanoparticles with very complicated shape, which can have different applications and just simple nanospheres. In order to prove that these particles are really gold, besides transmission electron microscopy, we used, of course, a straightforward physical instrumental technique. We used X-ray fluorescence analysis, XFA. This is a very typical technique. For those who apply know it, who don't, I recommend it very much. So the principle is that X-rays are applied to the sample, and the specific fluorescence from every element shows the elemental composition and even quantitative elemental composition. So you can, as usual, you can compare the line intensities, not that straightforwardly, but using some sensitivity coefficients, but some semi-quantitative analysis could be done. So in this case, for both this strain, which gave nanospheres on the surface, and mutant strain, which gave nanoflowers, we obtained the several lines of gold. These three lines, main lines of gold, are shown. These are the bands which are shown, and you can see some other bands here and here, the same bands, which refer to the usual microcomponents of the cell. This technique shows mostly the heavy elements, so metals, in principle metals and semi-metals. So in this case, these bands refer to iron, zinc, and other elements which are typically present in the cell. But gold is obtained in large quantities, so you can see this here. So, okay, I hope that, how much time do I have?
Oh, you have still...
Half?
No, it's
close.
The first part.
The first part, yeah,
so
great, great. So let's turn to the reduction of selenium anions. Selenium is a very important microelement. There are places on the Earth where selenium is in deficiency, and a lot of illnesses, diseases are caused by deficiency of selenium, both in animals and in the humans. There are places where selenium is as a toxicant in higher amounts in soil in particular, or in water, and in this case some bioremediation activities should be applied, as far as selenium is not a metal, it is not that simple to fix it in the soil or to get it out of the soil. Moreover, selenium forms selenates with this oxidation state of +6, and selenites with this oxidation states of selenium of +4, and they are very well soluble in water. This means that these toxicants can migrate in the soil and aquifers unpredictably. So something should be done about that, and some ways of bioremediation of these substances should be looked for. So this is the essence of what I have said. So it is called an essential toxin. So in micro amounts it is necessary. Like most microelements, in higher amounts it can be toxic to organisms. And what is very important, selenium is a constituent of a number of enzymes in almost all living systems. The most frequent biological forms of selenium is selenocysteine, is one of the amino acids, so-called 21st amino acid, and selenomethionine, very important amino acids as well. Mostly selenite and selenate are toxic, and they are straightforward use for addition to, for example, to complement the selenium deficient fodder for animals is not that good. Sometimes the farmers prefer some organic forms of selenium, but this is another question. So as concerns this bacterium, although it is very well studied, we found that there are no data on the relationship of this bacterium to selenium. There are no information on the effect of selenium compounds on this bacterium. So of course, we tried to fill this niche, to fill this gap, and to find some information about that, to get some information. So we used 2 strains of this bacterium, as I have said before, anaphyte and endophyte. And the aim of this work was to study the effect of selenite, selenium oxidation of +4. I should say we also tried selenate, but the bacteria do not reduce selenate. This is another question. So I will stick to selenite. This is also a very important and relatively steady compound which is soluble in water. This is like sulfite, which is used in photography, which is a very typical substance. So the salt is like that. So the concentrations of selenium +4 we used were in this range. A very simple but very illustrative figure. First of all, this is a negative control, a cell-free control. So we took this salt without any cells and stored it for many days, so nothing happens. This is relatively stable, so the solution remains transparent and colorless. This is, so to say, a simple control. Cells grown in the absence of added selenite. So this is colorless and not that transparent, but because this is not a solution, this is a suspension of cells, but this is colorless. As far as we added these concentrations in millimolar concentrations of selenite, you can see that the red color appears in a few days, after 4 days already, and it still increases if more days are passing. So you can see that this range could be used even as a semi-quantitative test for color, a simple color test for the presence of selenium. Red color is a typical color of amorphous red modification of selenium. But I should say that there are a lot of examples of bacterial reduction of selenium forming elementary selenium, but not all of the bacteria reduce it to the nanoparticle form. So it could be red, but it could be amorphous, not nanoparticle, but just amorphous selenium. In this case, we tried to make some experiments, for example, the same transmission electron microscopy. So this is another example. The previous one is with liquid medium. We also tried it with the semi-solid medium on agar. So you can see that this is a control without selenium, and with selenium, you could see this is very important because you could see that selenium is found in those places where the colonies develop. This may mean that the selenium particles, elementary selenium, is related to the cells, but whether they are out of the cells or inside the cells, it should be studied also. So this is an enlarged view. We used transmission electron microscopy and a similar technique attached to it, electron energy loss spectroscopy, to derive, so to say, elementary map of selenium distribution across the cells. So here you can see a transmission electron microscopic view of a cell, and these electron-dense particles inside the cells and also in the cytoplasm are selenium nanoparticles. And electron energy loss spectroscopy, which show the red color inside the cells, show that selenium is mostly localized inside the cells. So every information is here. We also used X-ray fluorescence analysis to show that this is really selenium, not other particles. So you can see here. So the maximum intensity is normalized to the maximum amount of any element in the cell. So with no selenium, the maximum line is for zinc and iron and copper, which are typically present in the bacterium. You can see that the background is also high here because the amount of zinc, for example, is not too much. When we add selenite and grow the bacteria in this medium, we obtain the biomass, which showed here look, the traces of selenium which are present in every cell as well as a microelement is like this as compared to zinc. Here, after growing with selenium, we have two lines of selenium here, and here is zinc. You can compare these two lines of zinc according to the background. The background here is like that. Here is zinc, and here is selenium. So the selenium accumulation, this was the X-ray fluorescence analysis from the nanoparticle, which is shown here by the arrow. So in this case, you can see that selenium is readily accumulated inside the cells when the cells are grown in the sub-millimolar concentrations of selenium. Not everything is seen. So these lines typically correspond, these are two main lines of selenium which correspond to X-ray fluorescence analytical technique for this element. What is the general origin of these selenium nanoparticles, moreover inside the cell, which should show that the bacteria reduce selenium according to their metabolic activity? So we made a screening around the possible enzyme systems in the bacterium, and we found two which could, to our knowledge, of course we looked for the literature about that, two enzymes which could be involved. This is a straightforward selenate reductase. However, selenate was not reduced by this bacterium. So far, we guessed that this is not selenate reductase, which should be involved, but something else. We found a nitrate reductase, one of the enzymes. Why we thought that this enzyme could be involved in reduction of selenium? First of all, there are literature data for other bacteria which show that selenite anions may be reduced to selenium, to elementary selenium, not necessarily in nanoparticles, involving nitrate reductases. The chemistry beside this is the following. It is rather simple. Here you can see the structure, the overall structure of selenite anion and nitrate anion. They are structurally similar, although the overall charge of the anion is different. They are similar. Their structure is very similar, And there are examples in the biochemistry of bacteria when a very similar substance, structurally similar substance, is involved in the enzymatic activity of transformation of another substance which is just structurally similar. In this case, this probably takes the place. This is a selenite or nitrite ions. They are very similar, and therefore a possible mechanism for the selenium reduction, selenite reduction, may involve the inclusion of selenite ions in the already existing metabolic pathway of denitrification process. This process is very well studied for the bacteria of Azospirillum bacteria. So they denitrificate nitrate to ammonium or to elementary nitrogen when an excess of nitrate ions are in the medium. These bacteria are very active in the sense of nitrogen metabolic transformations, both from fixing of atmospheric nitrogen to the opposite process, to denitrification. So in this case, this denitrification process can involve selenium, selenate ion for the reduction of selenium. And of course, we tried to find an additional evidence for that. We took this kind of very interesting mutant strain. I told you before that this mutant is very interesting, and it is so not because it has a complicated and modified surface structure, but it is also impaired in denitrification process. It is impaired in the synthesis of just the enzyme which we look for, the nitrate reductase. And we found these are not yet published results in our laboratory. That is why I just mentioned them. But we found that this strain does not reduce selenite, although this is almost the same strain as the parent strain SP245, just impaired in the synthesis of this nitrate reductase. So this is an additional evidence that this enzyme is involved in the reduction of selenite. Of course, some more straightforward experiments should be done to prove this, but this is an illustration how it can happen in nature, and this can be, to our opinion, this route of reduction of selenite may be not the way of detoxification of selenite, but this is just a parallel route of denitrification process in which a parallel structurally similar anion is involved. So I'm finishing.
You have still 5 minutes.
This would be very nice to have questions. So I'm finishing already. As I told you, this is a relatively simple talk, and I hope that it would provoke some thoughts and some questions as well, maybe afterwards. So I would be happy to answer them. So the straightforward conclusions. First of all, we found for this very well studied, very widely studied bacterium that for the first time that it is capable of reducing gold to gold nanoparticles of different shapes, as I told you, related to the different structure and composition of surface of this bacterium, and also reduction of selenite, selenium in the oxidation state +4 to selenium in the form of nanoparticles. I should say that these selenium nanoparticles have a very wide range of applications, from semiconductor devices to agriculture. So it is very important that adding selenium nanoparticles to the fodder for animals, we can regulate the selenium metabolic activity in animals. So these nanoparticles are metabolized in the quantities which are exactly needed for the animal. If you add organic, dissolvable, and more metabolically active substances, or still more inorganic anions, if you add them in a more quantity, it could be toxic to animals. And you cannot sometimes predict how much they eat and how much selenium they take. But if you add these nanoparticles, their metabolism regulates itself. They do not take in more than they need. This is probably related also to some enzymatic activity, but this is a very important trick from the agricultural viewpoint, from the animal breeding. So this is one of possible applications of these nanoparticles. I tried to show you that different mechanisms were found to be involved in the reduction of gold from one side and selenium from the other hand. So cell surface structure was found to regulate the morphology of gold nanoparticles, which is also very important from the possible applications of green synthesis of such nanoparticles. The bacteria were able to reduce selenite, but not selenate. We should study why further. And elementary red selenium with the formation of selenium nanoparticles in these ranges of diameter were formed. So these plant growth promoting rhizobacteria could be used probably. It also should be studied and confirmed. Could be used as biofertilizers for phytoremediation or bioremediation of selenite contaminated environments. And finally, the ability of azospirillum to synthesize these nanoparticles can be of interest, of general interest to nanotechnology and in particular to nanobiotechnology. Very different applications could be found. So I think my main collaborator, Dr. Anna Tugarova from my laboratory, we have been, this is my former PhD student, and now she's working on her Doctor of Science dissertation. We make a lot of very good experiments together. Of course, some young people from time to time are involved in our research. A few grants which supported this project could be acknowledged. Thank you very much for your attention, and I ask you to pay attention to this view of the Volga River. This is a bridge over Volga near Saratov from the city of Saratov, which is over here. Saratov is rather large. It is about 1 million of inhabitants. There is a Saratov branch of Saratov regional branch of Russian Academy of Sciences. We have several academic institutes in Saratov. In one of them I work. This is the narrowest point of the Volga River, which is 3 kilometers. And what you see here, this is not another bank. This is a large beach, sandy beach in the middle of the Volga. So you can go here. The buses stop here. You can have a swim. The Volga is very clean now. And in the sandy places, you can see that the water in summer is 3 to 4 meters transparent, like in the sea. So diving and underwater hunting is developing now. So everybody is welcome to Saratov to see the Volga, to have a swim, and to collaborate if possible. Thank you very much.
And now we have more than 5 minutes for comments and discussions. So let's take advantage of the presence of experts in biochemistry and
let's think so.
I see we have some, maybe you will start.
Is it difficult to separate the nanoparticles from the dead bacteria that make them? Is it difficult and expensive?
You mean from the cells?
Yes.
You know, sometimes this is not that difficult. As far as nanoparticles have been formed, they have different properties. So sometimes even simple ultrasonic shaking of the system is enough to separate them, or you could use some enzymatic decay of organic part, I mean the cells or cell debris, to separate the metal or elementary particles. So actually, this is a technical part which is not very complicated. There are well-developed routes to separate nanoparticles from the cells. Although when the nanoparticles are inside the cells, like the case, as I showed you, like the case with selenium, this is maybe a bit more complicated, but it is not very, very complicated.
Last one. Maybe Lady was first, so please.
I have two questions. First one is, as I think you work in lab, and it was no really conditions like in culture, yes?
You're right.
Okay. So the question is, you showed that mechanism of formation, this nanoparticles of selenide, yes, selenium, is similar to formation of nitrogen fixation, yes? Nitrogen fixation in...
Not exactly, but denitrification, the opposite process. Okay.
Okay. So did you try to experiment, try to study if we have a condition of mixed this selenium and selenide ions and nitrogen ions, and how did they compete by speed if they should be in a real condition of life?
I have been waiting for this question, the excellent question. The question is exactly on the point. So I didn't speak about that at all, and I was thinking whether maybe somebody from the audience would ask this question. Thank you very much indeed. So this is a very interesting question. This is a question of parallel reactions, enzymatic reactions, and this is very important in biochemistry, especially if we study the environmental processes. Of course, the medium in the soil, for example, where these bacteria occur, is much more complicated than any medium which we make in the laboratory. So I think that I will not give you a direct answer. As I told you, we have just started these investigations, and the first results showed what I have shown you. But I think that it should be specifically studied with relation to the rate of those processes. So, of course, if we have much more nitrate in the soil, nitrate in the soil than selenium, probably the most reaction would be occurring with nitrate. But you know, usually nitrate is not that much in the soil, except if the soil is very polluted. Usually the concentrations of nitrate are also very low in the agricultural soil. So this means that I think that the competition of these two processes would be not very strict. At least the rate of the processes, of the biochemical processes, should be studied in order to compare the real system, in order to model the process in the real system, which contains both nitrate and selenite. Then we can answer just by calculations, using the rates of the processes. As far as we don't know the rates, the rate constants, we cannot say anything. This should be studied. This is a way to other experiments. But anyway, thank you for the question.
And one little question more. I don't understand rate. So when this selenide nanoparticles is formed, in which way this oxygen of ionic form released by bacteria? I don't understand it here.
I believe that the chemical route is much more complicated, so you needn't expect that oxygen is evolved as elementary oxygen. By the way, the fact that nanoparticles of selenium are found inside the cells is an indirect proof that the reduction of selenite is involved in the denitrification route, because denitrification involves the transformation of nitrate into the cell, transfer of nitrate into the cell, and then enzymatic reduction of nitrate to ammonium, not to nitrogen, to ammonium, inside the cell. Of course, this is this process which needs several electrons per one atom of nitrogen. It is a multi-step process. It is a complicated enzymatic step. But I think that the involvement of intracellular selenate, which is transformed into the cell, is involved in this process. This is also a parallel way occurring inside the cell. And then if nitrogen or ammonium is in the dissolved state, selenium is not soluble, and it forms, in some days, it forms nanoparticles inside the cell. But as I cannot say anything about the roots of transformation, because this is a very complicated process. We simply have no time for it.
Okay.
But this is inside the cell, it's for sure.
Thank you very much.
So maybe now is your question.
Thank you, Prof. Kamnev. It's very interesting reports. But my question is very deal with last questions of these ladies. What, in your opinion, where is the bacteria concentrated the metal? Into the cell or on the cladding?
In what?
Where,
The last word.
where it's the bacteria concentrated the metal? Inside the bacteria? Inside the...
You mean selenium or gold?
No, gold.
Gold.
Gold. Or on the cladding of the bacteria?
Well, we noticed in good transmission electron microscopic images that gold nanoparticles are covering exactly the cell wall. So they are from the outer part of the cell wall. So these are gram-negative bacteria. They have a very complicated cell wall, and the outer layer consists mostly of some polysaccharides, mainly polysaccharides. Some of them are acidic, some of them are reducing polysaccharides, and gold nanoparticles. So if you see a large view of a cell, nanoparticles sometimes cover exactly the outer part of the cell. Even our scientists who first observed it, they said, ironically, like a joke, that isosperillum, as every lady, isosperillum likes golden things to wear.
The size of your bacteria is 1 micrometers.
Yeah.
And the size of nanoparticles differs from some tens of nanometers
yes.
until some hundreds nanometers.
Yes.
How they take place?
So 1 micrometer is 1,000 nanometers.
Yes.
So 1,000, and if you have 10 or 20 nanometers, you can see those nanoparticles around the cell.
So the last short question, because we are...
So like small dots, like small dots.
I think that there are some proteins which play main role in the metabolism of the bacteria, and this protein take the metal to this structure.
In this case, gold may be reduced not only by proteins, but just by polysaccharides as well. Okay, and they cover, sometimes when gold is in large amount, they cover almost all the surface. This is the point. I didn't speak about that because of lack of time.
Okay.
This is an interesting question.
Are there other techniques of synthesizing such nanoparticles? And what are advantages of the presenting technique before them?
Of course, there are a lot of chemical ways of similar nanoparticles could be formed. The old citrate techniques, borohydride techniques, but usually all of them need some specific preparation, some specific time, and what is more important, in order to obtain the monodisperse particles, sometimes some specific ways should be developed for chemical synthesis. For this kind of green synthesis, maybe the main advantage is sometimes that knowing the ways of transformation of the element, like I showed you when some nanoflowers could be formed, maybe this is much easier to find these ways of synthesis than to find some chemical ways, because they are not that easy to obtain such kind of nano hedgehogs, so to say, so nanoflowers they are called. In this case, they are obtained in one step. The bacteria itself forms it, and this is important from this viewpoint. But of course, they are competing, the chemical ways and the green synthesis. But green synthesis sometimes appears to be more straightforward and easier.
Okay, thank you. We have to stop the discussion. Thank you very much once more for this interesting talk.